Vaccines have been successful in preventing many viral diseases, but provide little or no therapeutic effect in those who are already infected. Also, there's some major viral diseases, such as HIV, for which it's been impossible to produce a vaccine, at least so far. Most viral diseases, with the exception of those caused by human immunodeficiency virus, are self limited illnesses that do not require specific antiviral therapy. The problem is that by the time the individual feels ill, viral replication is completed and infected cells are being cleared. Antivirals must be given early in the infection or prophylactically to populations at risk. It takes time to identify the specific virus. There are no broad spectrum antivirals currently available, and to obtain and dispense the prescription antiviral drug. It's difficult to get the drug in time to be useful. Lack of diagnostic reagents that can rapidly give you a result has hampered the developing and marketing of antiviral drugs for many acute diseases, despite the existence of effective therapies. The arsenal of antiviral drugs is small, and most are directed against HIV and herpes virus. The primary reason is that an antiviral must be safe. Because viruses are intracellular parasites, compounds interfering with viral growth often have adverse effects on the host. Also, antivirals must be very potent. Even modest replication in the presence of an inhibitor provides the opportunity for resistant mutants to prosper. Many medically important viruses cannot be grown readily in the laboratory, such as hepatitis B and hepatitis C, and some have no animal model of human disease, such as measles and hepatitis C. The testing and development of safe, potent, and efficacious compounds against these viruses is difficult. And again, many viral infections are acute, so antivirals are really not that useful. Structure-based drug design is now a common method used by the pharmaceutical industry to identify a lead compound and take it forward for further development. Once a firm grasp of the biological process under study is attained, an array of candidate drugs can be rationally designed to interfere with that process. The safety and efficacy of the most promising candidates would be initially examined in various animal models. But to ensure these findings hold in humans, a few lead candidates would be trialed through three distinct phases that address toxicity, potency, and reproducibility. Fulfilling all previous criteria, an expert regulatory body would scrutinize the clinical trial data and decide on approving a promising therapeutic. Antimicrobial drugs are one of our most important tools to manage infectious diseases. Since the discovery of penicillin in 1928, hundreds of compounds have been developed and saved millions of lives. Despite this vast diversity of over 200 human viruses being known, surprisingly, only 90 drugs have been approved to treat viral infections. Nearly half of these target HIV. So far, none have been approved to fight coronaviruses. Most antivirals try to shut down specific steps of viral replication. However, viruses are obligate intracellular parasites that rely on their host cell to replicate. As many stages of both the viral and cellular life cycles are shared or are very similar, it can be difficult to identify compounds that can inhibit the virus without harming the cell. So, for instance, the example I always use is that you can use 10% bleach to kill HIV, for instance, to sterilize syringes and so on, but clearly that cannot be used in patients. Additionally, viruses may rapidly evolve resistance to a drug. So, how do scientists develop new therapies against viruses? 3 decades ago, most of the first discoveries of antiviral compounds were fortuitous, since molecules originally developed for other purposes were selected as antiviral candidates based on their success in other medical disciplines. These methods for antiviral discovery were empirical, and most of the time, the biological mechanism behind the observed antiviral effect remained unclear. A common first step now is to use compound screening libraries to find candidates that stop the virus from growing in cell culture. Anywhere from a handful to hundreds of thousands of natural and synthetic chemicals are tested for antiviral activity and toxicity. The U.S. National Institutes of Health maintains a library of over 350,000 compounds, and most large pharmaceutical companies have their own chemical databases for this purpose. After a lead candidate is identified, the next step is to test it in an animal model such as a mouse. Cells in a dish do not always behave the way they do in an animal, and so many drugs that may initially appear promising may fail to resolve the infection, or worse, may actually prove to be toxic to the animal. This is a vital step to ensure that new compounds appear to be safe and effective before they are tested in humans. Bioavailability refers to the extent and rate at which the active moiety, the drug or the metabolite, enters the systemic circulation, thereby accessing the site of action. Pharmacokinetics defines what the body does to the drug. Pharmacokinetics is the study of a drug absorption, distribution, metabolism, and elimination from the body. These are important determinants for the success of a new drug. The late-stage attrition of chemical entities in development and beyond is highly costly, and therefore such failures must be kept to a minimum by setting in place a rigorous, objective quality assessment at key points in the discovery process. This assessment needs to begin as early as possible and must be of high stringency to prevent precious resources being squandered on less promising leads. The earliest point at which such knowledge-driven decisions can be made is in the lead generation phase. Here, the initial hits are progressed into lead series by a comprehensive assessment of chemical integrity, synthetic accessibility, and other properties. Optimization of the ADME, absorption, distribution, metabolism, and excretion properties of the drug molecule is often the most difficult and challenging part of the whole drug process. The ADME profile will also have a major impact on the likelihood of success of a drug. The early awareness of the required profile, a given selectivity, solubility, permeation, metabolic stability, and so on, is important for the selection and prioritization of series with the best development potential. In this regard, it is important that at least 2 lead series of significantly different pharmacological and/or structural profiles are advanced as reserve or backup lead series. This ensures against failures due to unpredictable factors such as toxicological findings in later animal studies. The effect of such a rigorous process at an early stage is to achieve greater awareness of key liabilities, which can be addressed in adequate time and with sufficient resources. The net effect is to reduce attrition in the costly clinical phases by intercepting many crucial ADME-related issues before they are discovered too late to be resolved. The cost of bringing an antiviral drug to market can easily exceed $100 to $500 million. Thousands of leads may yield only one candidate for further testing. Research and lead development, the R in R&D, is only the beginning of the process. The D is safety testing, scale up of synthesis, formulation, pharmacokinetic studies, and clinical trials. All of these kinds of studies usually take about 10 years after the initial lead is identified. More promising leads are discarded due to toxicity than to any other factor. So when we look at this, we look at a cytotoxic index, and we're hoping for a ratio of 1 to 1,000 or smaller. After introduction into a person, the drug must reach the proper place at the proper concentration for an appropriate time. It's best if the drug can be marketed as an oral dosage. That's going to be easier to get out to the patients and far cheaper. Ability to manufacture and market a drug at an acceptable cost is key. If a drug is exorbitantly expensive to produce, then it's not going to succeed as a drug that can be marketed. So now we're going to talk about critical points for drug hunters. First, we'll consider virology issues, then we'll consider business issues. Under virology issues, the most important, of course, is proof of principle. What is the molecular mechanism? Does the drug work in cell and animal models as predicted? The compound should block viral spread early to limit cytopathology and host response. This is key. The drug must be potent and block replication completely. Otherwise, it could be making an infection persistent by merely slowing replication and spread rather than stopping it. Resistance is an issue as well. We've already talked about how the mutation rate in viruses is very high, particularly in RNA viruses. So the generation of mutants is a guarantee. Resistance mutations arise when any virus is permitted to replicate. Antiviral compounds select for these spontaneous mutants. Viruses resistant to other drugs must not be resistant to your drug. Noncompliance by a patient may select for drug resistance or may encourage persistence. Dosing, dietary requirements, unpleasant taste, or side effects are all factors that can result in noncompliance by a patient. So this is something that has to be considered. Then there are toxicities. Long-term human toxicology studies are difficult and expensive, but short-term toxicology studies may not predict side effects arising when drugs are taken over an extended period of time. Now, if we move on to business issues, the compound, of course, has to be safe with no side effects. That's obvious. Also, the compound has to be inexpensive to manufacture. As we mentioned before, if the drug is extremely expensive to produce, then it's not likely to be developed into a commercially available drug. So it has to be easy to formulate and to deliver. One thing that you might not think about is that the compound must satisfy an unmet medical need. You don't want to spend years and millions of dollars producing a drug that's going to be competing with an existing effective drug. So you want to make sure that you're not just taking care of a problem that already has a drug to provide a solution. Also, the disease severity plays a role. How severe is the disease? Cold versus Ebola. We're going to be much more interested in coming up with a drug that can treat a viral disease that has a high mortality rate. And then, of course, the profit has to be possible. which takes in all of these kinds of factors. But there is some talk about the FDA playing a bigger role, marketing drugs to patients themselves when a cheaper one is already available but may not be under patent. So there's all kinds of issues that come to play in developing drugs. It's not just finding a drug that works. There's all kinds of issues, and money does play a role in all of this. The first step of viral replication is an attractive target because virus receptor interactions offer promise of high specificity. However, you can't interfere with the normal function of the cellular receptor. Remember, the receptor on the cell for a virus did not evolve to provide entry for a virus. It serves some other function for the cell. And you can't be interfering with that, or it could be cause toxicological problems. Also, this assumes that the virus has no other mode of entry. Many viruses have alternate receptors. Some studies have looked at blocking fusion. For instance, 2 anti-flu drugs block uncoating. Various viral proteins have provided good targets for antivirals. Viral messenger RNA typically differs from host cell messenger RNA. The viruses use this to its advantage. Translation of host cell messenger RNA is often diminished within a few hours after infection, while viral messenger RNA is proceeding rapidly. So the idea here is that you might be able to develop a drug that can exploit this. The replication of viral nucleic acids is also a target. Many viruses require an enzyme not found in uninfected cells. It should be possible to design specific inhibitors. We all know an example, AZT. Acyclovir goes after the viral DNA polymerase of HIV. Integration, morphogenesis both require special enzymes, proteases, and so on, that are not found in uninfected cells. So these are all targets, and there are drugs out there that target each one of these different steps for various viruses. And they're drugs either already developed or in development that attack these particular steps. But it's all part of trying to identify a way that you can target a viral activity without hurting cellular activity. If a drug is being used, either repeatedly in a population or long-term in a patient, you can expect the development of resistance. And when you have the development of resistance, we talk about a virologic breakthrough. This is just something you have to expect. So a key part of drug development is finding ways to minimize resistance. One way that you can do this is to use multiple drugs in combination, either together or sequentially. This is what's done with the strategy called HAART, highly active antiretroviral therapy that's used for AIDS. They're using a combination of at least 3 drugs, and that's able to prevent the development of mutants that can survive. You can also restrict the use of your antiviral drug to situations where there is a clinical need. This is similar to what we currently do with antibiotics. Antibiotics for many years were over prescribed for different kinds of conditions that didn't really require antibiotics. Now doctors are much more careful about prescribing and using antibiotics in their patients, and that's all about controlling resistance. Also, you want to prevent widespread veterinary use of drugs that are required for human therapy. The U.S. bans the use of any anti-influenza drugs in animals so that the drugs will remain effective for humans. Now, there are far fewer antivirals than antibiotics, so preventing the development of resistant viruses is even more critical. The major mechanisms for drug resistance are mutations in the viral DNA polymerase gene, or in genes that encode for the viral kinases required for the activation of certain drugs, such as acyclovir and so on. Widespread antiviral resistance has limited the clinical utility of M2 inhibitors for the prevention and treatment of influenza infections. So let's take a look at the antiretroviral therapy that's used. People who are on this, sometimes it's referred to as HART, sometimes just ART, but people who are on this take a combination of HIV medications every day. And this is all based on the fact that HIV mutates at a very rapid rate. Shortly after the discovery of HIV in 1981, a variety of therapies using one drug were introduced into people living with HIV. This included the drug AZT. Despite initial success, these monotherapies proved to be ineffective at slowing the progression of the virus. The failure was due to HIV's ability to quickly develop resistance to these single drug treatments. In other words, HIV mutated or changed into a form that no longer responded to the individual drugs. In 1995, a combination drug treatment known as the AIDS cocktail was introduced. This type of therapy is now known as the highly active antiretroviral therapy, or HART. It's also called combination antiretroviral therapy, or just simply antiretroviral therapy. But the idea here is you don't use just one single drug, or the monotherapy. You use multiple drugs. That means that any virus that mutated would be so badly damaged that it wouldn't be able to survive and replicate. And so far, this strategy has been very effective. We have the problems with resistance developing in influenza drugs also. Two older drugs, amantadine and rimantadine, had been approved for treatment and prevention of influenza A, but many strains of influenza, including the 2009 H1N1 influenza, are now resistant to these drugs. CDC has not recommended the use of these drugs for recently circulating influenza viruses, although recommendations could change if there were future reemergence of specific virus strains with susceptibility patterns favoring such use. Peramivir is a neuraminidase inhibitor that acts as a transition state analog inhibitor of influenza neuraminidase and thereby prevents new viruses from emerging from infected cells. Zanamivir works by binding to the active site of the neuraminidase protein, rendering the influenza virus unable to escape its host cell and infect others. Tamiflu is an antiviral medication used to treat flu symptoms caused by influenza in patients who have had symptoms for less than 2 days. Tamiflu may also be given to prevent influenza in people who may be exposed but do not yet have symptoms. So this drug, Tamiflu, was approved for medical use in the U.S. in 1999. It was the first neuraminidase inhibitor that could be taken by mouth. And the U.S. has stockpiled a large supply of Tamiflu to break out in the case of any sort of pandemic of influenza that could arise. The San Diego firm MAP Biopharmaceutical leaped into national news in 2014 when its experimental medicine, ZMapp, was used during the West African Ebola epidemic. The drug is a mixture of 3 antibodies meant to knock down levels of the Ebola virus in the body, hopefully buying time for the patient's immune system to rally and defeat the disease. Anecdotal accounts indicated ZMapp might have helped save the lives of infected patients, such as the African missionaries Dr. Brantley and Nancy Wrightbull, who were U.S. citizens that came down with Ebola. ZMapp spiked in demand during the 2014 outbreak, but the company wasn't set up to make its drug at mass scale back then. By early 2015, when a clinical trial was finally authorized to start treating patients with ZMapp, the epidemic was waning. The study moved forward anyway, but had a hard time recruiting patients to test. In the end, the data showed some evidence that ZMapp helped Ebola patients survive, but not enough patients were enrolled in the trial for the drug to prove statistical significance. Since the commercialization of the first therapeutic monoclonal antibody in 1986, they have represented the largest and fastest growing class of biopharmaceutical products. The development of monoclonal antibodies has revolutionized the pharmaceutical industry, creating a multi-billion-dollar market, and it's provided vast opportunities to treat a wide range of diseases, from autoimmune disorders to cancer to viral diseases. Monoclonal antibodies produced in mammalian cell culture have achieved remarkable pharmaceutical and financial success, leading to several blockbuster therapeutic products. However, they are prohibitively expensive, making them unaffordable for the majority of countries around the globe. Monoclonal antibodies produced in mammalian cell cultures requires capital-intensive facilities, fermenters, expensive downstream processing, cold storage and transportation, and sterile delivery methods. The high cost of this system has encouraged the development of alternate production systems, including yeast, insect cells, and plants. While there is much progress being made with these systems, the general public has very little knowledge about them and their utility in producing monoclonal antibody drugs for human use. We'll talk more about monoclonal antibody drugs at the end of this lecture when we talk about the treatments that President Trump is receiving for coronavirus. Plants, especially tobacco and its relatives, have been used to produce MABs for decades, with the first MAB made in tobacco in 1987. Despite the complexity of MAB production requiring the assembly of 4 polypeptides into a heterotetramer in complex glycosylation, a variety of MABs and their derivatives have been produced in many plant species, such as secretory IgAs, tetravalent MABs, immune complex, single-domain fragments, single-chain variable fragments, and diabodies. An increasing number and diversity of MABs are being produced every year, and several of them have now reached human clinical trials. The traditional advantages of utilizing plants to produce MABs are their demonstrated low cost, high scalability, and increase safety, while a major advantage of plant production systems is their low-cost upstream protein expression. In contrast to mammalian cell culture systems, which require capital-prohibitive bioreactors and expensive culture media, plant MAB production can be easily scaled in inexpensive greenhouses. Tobacco plants can be grown in soil or hydroponically in simple mineral solutions to produce a large amount of biomass and millions of seeds per plant for an easy scale-up of production, making high levels of MABs efficiently and sustainably. Plants rarely carry human or animal pathogens, reducing the risk of contamination. In addition, plant cellular protein machinery can easily assemble multiple subunits required for the production of MABs and efficiently perform critical post-translational modifications. New innovations in viral vector development provide additional benefits for plants to make MABs. For example, the development of the deconstructed viral vector system has successfully addressed the challenges of sufficient protein expression levels consistently and speed of MAB production in plants. In contrast to using stable transgenic plants, transient expression with deconstructed viral vectors avoids the month-long process of generating and selecting transgenic plants. It can produce up to 5 milligrams of MAB per gram of fresh leaf weight within 2 weeks due to its robust transcriptional and translational activities of the plant virus components in the vector. These deconstructed viral vectors are non-competitive, allowing MABs and other proteins with more than one heterosubunit to be expressed and assembled in one individual cell. Agrobacterium tumefaciens, a bacterium that normally transfers a portion of its plasmid into plant cells, is used to deliver the deconstructed viral vectors to plant cells. Thus, the deconstructed viral vector system provides the flexibility of nuclear gene expression with the speed and high yield of viral vectors. The rapid and high-level MAB production capability of the transient expression systems makes them the system of choice for obtaining the milligram and gram levels of MABs needed for preclinical studies. These vectors have also been adapted for scale-up manufacturing of MABs in stable transgenic plants while retaining consistent and high MAB expression level and transgene stability. Therefore, rapid evolution of MAB candidates and transition to a large-scale commercial production can be accomplished by the combination use of both transient and stable plant expression based on deconstructed viral vectors. Let's look at the drug development for hepatitis C. Hepatitis C is the #1 cause of liver cancer and liver transplants. It's brought on by a virus you can catch if you come into contact with contaminated blood. You could get it from an unclean tattoo needle, for example. Sometimes it spreads during sex. For decades, you needed painful shots of a medicine called interferon and a pill called ribavirin. These drugs didn't target the virus that made you sick. Instead, they amped up your immune system so you'd fight it the way you do when you get the flu. But the treatment didn't always get the virus out of your body. Cure rates hovered around 50%, and people who stuck with the year-long treatment, and not all did, had to live with chemo-like side effects. So it wasn't a pleasant thing to go through. These days, more and more people can get rid of the virus simply by taking a pill at home for just a few weeks. There are several ways to do it without having to get shots. Now, there's no one-size-fits-all option. There are many different types or genotypes of hepatitis C. Type 1 is the most common. This is important to understand when you talk to your doctor. Not all meds work on all types. Which medicine is best for a particular patient depends on how much liver scarring or cirrhosis they have. Hepatitis C treatments are highly effective in curing chronic infections, with newer generation drugs improving cure rates even amongst those who have previously failed HCV therapies. Typically, between 20 and 30% of HCV infections are spontaneously cleared by the patient with no medical intervention. Of the remaining 70 to 80%, the infection can progress over the course of decades and gradually damage the liver. About 10 to 20% of those infected develop severe conditions such as cirrhosis, liver cancer, or end-stage liver failure requiring a liver transplant. Treatment for hepatitis C depends on several factors, including how much virus is in the patient's body, their viral load, the genotype or strain of hepatitis C that the patient has, how much damage to the liver, do they have cirrhosis, and so on, what other health conditions does the patient have, and what has been the response to any previous treatments to hepatitis C. Doctors have to be careful when they use the word cure in the context of hepatitis C. It's a very complex virus, one that is proven to be highly resistant to treatment in the past. So understandably, clinicians are wary of oversimplifying or overpromising. Most often the term specialists use when they talk about successful treatment of HCV is sustained viral response, or SVR. A successful treatment means that hepatitis C is not detectable in the blood for at least 12 weeks after completing treatment. The question now is whether SVR is the same as a cure, and the answer has to do with whether or not SVR is permanent. Some doctors do use the word cure because SVR is as close to a cure as we can get with HCV. Others prefer to use the word remission because of their doubts about how permanent SVR really is. In reality, the most recent studies suggest that the likelihood of SVR being permanent is very high. Here are 2 points to keep in mind. Studies show that once patients have achieved SVR, they have a 99.2 to 100% chance of remaining hepatitis C free. Less than 1% of people who achieve SVR experience a return of hepatitis C infection. When HCV does return after achieving SVR, there's a good chance it is due to re-exposure. In effect, it is a new infection, just in the same person. Most commonly, this is seen amongst IV drug users, or prisoners, or those co-infected with HIV. However, even when taking these results at face value, the science of treating hepatitis C is relatively new. Several unknown issues remain regarding exactly how to eliminate this viral infection. Until these unknowns are clarified, many physicians will avoid calling SVR a cure, although there are many commercials now for the various hepatitis C drugs out there, and of course they're all calling it a cure. It takes about 10 years for experimental drugs to go from the lab to the medicine chest. First, we have the preclinical stages. Lab and animal studies are done to show if the drug is safe and effective. If a drug passes through the preclinical stages, then it goes on to 3 phases of clinical trials. Phase 1 establishes that the drug is neither lethal nor toxic, and it also looks at safe dosage ranges. This study would be done in healthy volunteers and last about a year. Phase 2 enrolls 200 to 300 patients, and you're comparing your drug to existing treatments or a placebo. Physicians monitor the patients closely to determine efficacy and to identify adverse reactions. This phase takes about 2 years. And then phase 3, which involves thousands of patients, measures your drug treatment against established therapy. This phase takes about 3 years. The FDA has been working since the 1990s to allow certain drugs to go through a faster development and approval process based on encouraging preliminary studies. There are 4 accelerated regulatory categories. There's fast track, there's breakthrough therapy, accelerated approval, and priority review. These categories vary in how they are applied, but their common thread is that they are meant to at least temporarily sidestep the randomized controlled trial part of the traditional approval process. In randomized controlled trials, participants are not told whether the treatment they are receiving is the actual drug or placebo. Randomization removes bias and helps to establish cause-effect relationships between an intervention and an outcome, so these kinds of trials are considered the gold standard for drug research. We just talked about the Coronavirus Treatment Acceleration Program, or CTAP, that was designed to fast-track SARS-CoV-2 drug approvals. Okay, so let's finish out this lecture talking about what's going on with coronavirus treatments. The FDA has created a special emergency program for possible coronavirus therapies, the Coronavirus Treatment Acceleration Program, or CTAP. The program uses every available method to move new treatments to patients as quickly as possible, while at the same time finding out whether they are helpful or harmful. They continue to support clinical trials that are testing new treatments for COVID so that we can gain valuable knowledge about their safety and effectiveness. If you look at the bar chart, there are several important categories. Antivirals, of course, are drugs that keep viruses from multiplying and are used to treat many viral infections, such as hepatitis C that we just talked about. Immunomodulators are aimed at tamping down the body's own immune reaction to the virus in cases where the body's reaction basically goes overboard and starts attacking the patient's own organisms or own organs. And we've seen this occurring in patients with COVID. Neutralizing antibody therapies may help individuals fight the virus and include manufactured antibodies, animal-sourced antibody therapies, and blood-derived products such as convalescent plasma and hyperimmune globulin, which contain antibodies taken from people who have previously had COVID-19. Cell therapy products include cellular immunotherapies and other types of both autologous and allogenic cells, such as stem cells and related products. Gene therapy products seek to modify or manipulate the expression of a gene or to alter the biological properties of living cells for therapeutic uses. The diversity of therapeutic approaches being investigated is important because it rapidly expands our understanding of the effect of different categories of potential treatments. However, we've got hundreds of drugs currently in development. We've had trials, hundreds of trials reviewed by the FDA. There are 5 treatments currently authorized for emergency use only, but right now there are no treatments approved by the FDA for use in COVID-19 without an emergency use approval. Okay, so just as I thought I was finished recording this lecture, we got the news about President Trump developing COVID. And I thought that it was important that I include a discussion about the treatments that he's getting. They're at this point in time when our president is sick, that's very significant, and particularly since it's a viral disease, I think that it's appropriate for us to discuss it in this class. So I made these new slides, and I was getting ready to record the last 3 slides when the announcement came out that a new drug had been added to his treatment. So I just tossed that one in too. So this is the latest news as of about noon on Sunday. So the president is receiving remdesivir and a polyclonal antibody cocktail treatment, and I have separate slides to go into some detail on those. But the drug that was just added this morning to the list was dexamethasone, and dexamethasone is a steroid. Now, just a couple of months ago, researchers in England announced that they had evidence that this drug could improve COVID-19 survival. And it has the advantage that it's a cheap, widely available steroid. Their data showed that it reduced deaths by up to 1/3 in severely ill hospitalized patients. They said that the study was large, and so over 2,000 patients, and they compared them with 4,000 patients who were getting the usual care. The drug was given either orally or through IV, and after 28 days, it had reduced deaths by 35% in patients who needed treatment with breathing machines, that is, respirators, and by 20% in those only needing supplemental oxygen. It did not appear to help less ill patients. So that leads to questions as to why they decided to add this to the president's treatment. Remdesivir was the first drug to get emergency authorization from the FDA for use on COVID-19. This drug is a nucleotide analog, and the triphosphate form of the drug is being used as a substrate for many viral RNA-dependent RNA polymerase, or RDRP complexes. Now remember, we talked about these early on. For viruses that use RNA as its genome, they have to bring their own polymerase to do this, because you're making RNA from RNA. And a eukaryotic cell does not have a polymerase that can do that. So that's what this drug is targeting, that RDRP complex. And it's been reported to inhibit viral RNA synthesis by a specific mechanism of delayed chain termination for all 3 coronaviruses, so MERS, the original SARS, and now our SARS-CoV-2. Remdesivir was originally tested as an antiviral against Ebola and hepatitis C, only to deliver lackluster results. But a randomized controlled trial published in May concluded that the drug reduced the recovery time of people hospitalized with COVID-19 from 15 to 11 days. The trial did not show any effect on mortality, though retrospective data released in July hints that the drug might reduce death rates among those who are very ill. The FDA responded to this data in May by issuing an emergency authorization for remdesivir's use in critically ill patients who need supplemental oxygen. In August, they expanded that approval after researchers found that patients with less severe forms of COVID-19 seemed to benefit modestly from a 5-day treatment course of remdesivir. The revised approval allows the use of the drug on all patients hospitalized with COVID-19, regardless of how severe their disease is. This move was criticized by some experts who said the FDA had expanded remdesivir's use without strong evidence to back the change. And then we have Regeneron. The monoclonal antibodies are clones of potent SARS-CoV-2 antibodies that can neutralize the virus, at least in test tube studies. Researchers plucked the genes for these antibodies from humans who recovered from COVID-19 or from mice artificially infected with the virus. The companies then put the genes in Chinese hamster ovary cells to bulk manufacture the antibodies, which were given to the COVID-19 patients as infusions. The target population for the monoclonal cocktail are SARS-CoV-2 infected people who have not yet mounted their own immune response and have exceedingly high levels of the virus. What they're trying to do is to turn these individuals into patients who have already started to effectively fight the virus. In the Regeneron data, the most dramatic drops in SARS-CoV-2 were seen in seronegative patients who had the highest levels of virus at the trial start. In comparison with patients who received the placebo, the results were clearly significant. Using its proprietary Veloc-immune mice, Regeneron screened thousands of fully human antibodies. The mice have been genetically modified to have a human immune system. They also evaluated antibodies isolated from people who have recovered from COVID-19. They then chose the two most potent, non-competing antibodies that were most effective at neutralizing the virus. They then scaled up the dual antibody cocktail for clinical use with the company's own Velocimab and manufacturing capabilities. Now go back to the audio on the ZMapp treatment and my discussion of the potential of plants for monoclonal antibody production. This picture shows the biotech production specialists working at the Regeneron Pharmaceuticals facility in New York. The large vats are full of cells that churn out the monoclonal antibodies. MABs produced in mammalian cell cultures, as the Regeneron antibody cocktail is, require capital-intensive facilities, fermenters, expensive downstream processing, cold storage and transportation, and sterile delivery methods. So it's a very time-consuming, detailed, and expensive process.